Yeah, thank you, Nuria, for the warm introductions. So my name is Jiyu, and I'm very glad to have this opportunity here today to introduce our recent studies and share my stories as a PhD student and probably an early career researcher. So the topic I am going to introduce for today is our recent publications to the ISMB 2022 titled with Exploring Automatic Inconsistency Detection for Literature-Based Geontology Annotation. Before getting started, I would like to introduce the outlines of my talk for today. So first, I will introduce what is literature-based geontology annotation and what makes it different from other types of Go annotation within the biological databases. Secondly, I will explain why literature-based geontology annotation requires automatic consistency assurance. And following with this explanation, I will introduce the challenges in the implementation of such kinds of automatic approaches, including the requirements that must be followed. And then I will extract some key details from our studies and demonstrate how we tackle those challenges. And finally, I will conclude with a discussion of opportunities for automating the Go annotation quality assurance from a broad perspective. So now we are ready to get started. So literature-based geontology annotation is very different from other types of Go annotation. It is the result of the manual curation, which means it is more reliable than any other types of Go annotations. They often have support of direct evidence, often written in natural language from the associated literature. Some publications may also call this as primary literature. So I will show a real example of literature-based geontology annotation later. These annotations have experimental type evidence code. So here I have listed the name and the acronyms of the experimental type evidence code provided by the Gene Ontology Consortium. So previously, the production of or the generation of the geontology annotation has been the focus of the research communities for decades. But until recently, the focus has changed to the quality assurance on existing annotations. So this is because some species, such as human or Homo sapiens, already have enough gene function knowledge. However, the consistency of this knowledge, such as being presented between the old and new records or publications, was badly maintained. So here I have attached the screenshot of two publications from the Go annotation research groups. They all mentioned the importance of maintaining existing Go annotation resources. Here is a broad summary of the current approach for quality assurance. It is manually conducted with the help of expert curators, and the curators are often assisted with semi-automated systems, such as the document triage system, to help find the relevant literature. The curation also involves collaborations from different communities. But the problem is this approach is inefficient and cannot scale up with the size of existing Go annotation resources. So the graph on the left-hand side shows the growth of the annotated scientific publications over time. These publications often require rereading or the re-review by the curators when they're conducting the quality assurance. And on the right-hand side shows the total number of annotations with experimental type evidence code. So these two demonstrated statistics are directly extracted from the Gene Ontology Consortium, the 2022 July release. And in terms of semi-automatic systems, unfortunately, these systems still lack of support regarding the reliability and the efficiency. So this is a cutting-edge system called PubDaters developed by ISCBacademy. So the system can automatically process a full text document and highlight the key concept mentioned in the raw text. The right-hand side shows a list of the gene mentions recognized or identified by the system pubdata. However, if we refer to a real-world database, only 5 genes are annotated by curators as ground truth. So this database is called Amigo. The precision, so we can see that the precision of such kind of system is very unpromising. So given these motivations, we set up our research objectives. We aim to develop a whole new automatic system that can take a Go annotation record as input, detect inconsistencies in the representation of gene function, and flag suspicious record in a typed inconsistency as the output. So on the right-hand side, I show a demonstration of a basic framework of such kind of system. So the system will rotationally retrieve the Go annotation record from the biological databases, and it will extract some major components, such as here we illustrate 3 components: the literature, the ontology term, and the gene product. And then some key informations will be extracted from these 3 components, such as the evidence sentence, the text attribute of the Go terms, and the normalized gene symbol. And then these information will be forwarded into a automatic inconsistency detection model, supported or backended by text mining techniques. And these models will flag suspicious records and forward them to curators for a manual curation. So unfortunately, the implementation of such kind of system is not easy. And here I have just listed 3 key challenges. For the first challenge is we found that there is no systematic studies on the characteristics of the Go inconsistencies. Existing publications are all discussing the quality issues or inconsistencies on a very broad level. So to compensate this, we formalized 4 major types of inconsistencies, and we assume that they confuse human and computer. The second challenge is we have no data. So to compensate for that, we design heuristic rules to simulate inconsistencies in the real-world databases and use that rules to generate a synthetic dataset. And the last challenge is currently there is no feasible approach have been implemented. So, and we are proposing a framework based on for the detection of inconsistencies. And we typically focus on 4 aspects. One is we make sure the framework is suitable for both rule-based and machine learning-based models. Secondly is it shall satisfy a human-in-the-loop application scenarios. And thirdly is it shall adaptive to the open world knowledge. So in futures, when the new species or new Go annotations is added into the databases, the system can still cover the quality assurance. And finally is we approve or sort of test the feasibility of this proposed implementation with empirical experiment and qualitative case studies. And now I will introduce the consistency validation involved with the Go annotation or ontology annotation. So I guess we're very familiar with the ontology annotations. It uses controlled vocabularies called Go terms to uniformly represent certain gene function knowledge. And in terms of a record level consistency assurance, it often involves with the validation of at least 3 components. One is the ontology term, the literature evidence, and the gene product. On the right-hand side, a table, I demonstrate 2 examples of such kind of Go annotation and show how they're consistent between the 3 components. For the first example, we can see the ontology term is negative regulation of feeding behavior. And this Go term is being selected to annotate a mouse gene called TRH. And the supportive evidence is on the right, which is, it says how the mouse genes have the function that has a negative regulation of feeding behavior. So that is a record level consistencies between the three components. The automatic assessment of consistencies in Go annotation is critical because one is it can prevent the cascading errors within biological databases. That means when one of the Go annotation have inconsistency issues, it may also spread that quality issues to other annotations. The second is it can help alleviate negative impact to the downstream tasks, such as the gene expression analysis. And thirdly, of course, because it is an automatic system, we hope it can improve the efficiency of database curation or database quality assurance. And of course, apart from the record level inconsistencies that we have just introduced, there are also many broad discussions of other types of quality issues in the biological databases, such as I have highlighted some publications here that discuss duplications, redundancies, inconsistencies, and biases. So we start with the study of tackling inconsistencies, and we hope that we can extend our studies to cover other types of quality issues in the near future. So, and now from this slide, I will start to introduce some technical details of our studies. The first is how we define different types of inconsistencies. In our current studies, we define 4 major types of inconsistencies. These are consolidated because they all violate the key quality assurance guidelines. And these inconsistencies are studied because they are hard challenge or code challenge to both human curators and computers. On the right-hand side is a geontology graph. I use this graph to show what is an over-specific selection of Go term and over-broad selection of the Go term. So for example, if the correct annotation or the consistent annotation is fitting behavior, then if a curator is selecting suckling behavior as the annotation, then that annotation will be considered as over-specific. And comparatively, if the curator is selecting behavior as for the annotation, then that will be considered as a broad selection of Go term. And there are also other 2 types of inconsistencies, which I will cover in later slides. So here it shows the over-specific selection of Go term I just mentioned. So on the left-hand side is a real example of the Go annotation. The gene is TRH. The evidence sentence is the one I just demonstrated in the table. The Go term, the correct consistent Go term is the feeding behavior, but the suckling behavior, which is the direct children of feeding behavior on the geontology graph. So this Go term is considered. If this Go term is annotated to the gene, given this evidence, then this entire annotation will be considered as over-specific. So this type of inconsistencies confuses both humans and computers. Another type of inconsistency, it is called irrelevant Go mention in the evidence text. It confuses computers at most. Like this one shows the word spindle is actually a Go term. It is mentioned as a keyword within the given evidence text. And also this evidence text introduce a gene product called mTOR. But if we read this evidence sentence, we can find that the spindle is not telling the cellular component information of mTOR. So it shall not be annotated to this gene. So but existing automatic Go annotation production systems, such as the puptators or concept mappers, they can recognize this concept as an entity, but they cannot distinguish whether it is truly relevant for a Go annotation or not. So that is why it is confused for computers. So, so after introducing the characteristics of inconsistencies, we have a new challenge. So first is we have challenges when accessing the data. We do not have enough available inconsistent data in hand. The reason we need that is because if we want to implement a machine learning supported language model to infer the inconsistencies, we need to feed the model with both consistent and inconsistent records. So the model can generalize the similarities and the differences between the two. So for a consistent instance, we have experimented with two different collections of data. The first one is a small corpus called BC4Go. Its consistency is guaranteed by expert curators with the annotation of evidence at sentence level. The second one consists of records directly sampled from resources called Jiyu provided by NCBI. It is mostly updated and rich in quantity, but contains noises. So the noises is, for example, there might be inconsistent annotations in the second collection of data, but were treated as consistent instances. So to solve this problem, we have designed a noise injection strategy to toggle the scale of noise or the degree of noise within the dataset and trying to analyze the impact of noise to the model's overall performance. So after having consistent instances in hand, we design heuristic rules to modify consistent instances to simulate inconsistencies. So here I demonstrate one of the rules for simulating over-specific type of Go annotation. So we refer to the Ontology knowledge graph and randomly select a child term to replace with the original Go term in the consistent annotation. And after doing this modification or replacement, we manually confirm that the modified instance, the annotation, the new annotation, does not exist in the current knowledge base to ensure that it is truly valid as a over-specific type inconsistency. So again, I'm using the same example on the right-hand side shows how does the over-specific inconsistency look like after the modification. So I will skip the introduction of like generating or synthesizing other types of inconsistency. And but instead I will move to the modeling part. So we must take into considerations of some primitives because we want to make sure that this automatic implementation can be adapted to human in the loop curation settings. So as I just mentioned, due to the limitations in creating noise zero training set or data set, we apply noise injection strategies into that. The idea is we flip a consistent instances into either type of the inconsistencies or vice versa as a way of generating the noises. And based on that, we build different models for dataset with various degrees of noises and to compare the performance on these different versions of the models. Apart from the noise, we also design studies for out-of-distribution instances. So it simulates when rare species or new species are added into the knowledge base. So these new database records are considered as out of distribution because they have never been seen by the models. We will see how does existing models can adapt to them. For example, we're training with human ontology annotation records, but testing that on Go annotation records of rat species. And again, this is a re-emphasize of the framework. And overall, we have to ensure that the system is moderated by human curators. So you can see that in the major part of the systems, we have a manual curations in the middle of the loop. So from the next slide, I will start to introduce some technical details of the automatic consistency detection model. So firstly, in order to encode the Go hierarchical knowledge, we designed a two-layered graph neural network models with the objective of edge type prediction. So there are 4 different edge types, is a part of are the relations in the geontology graph. And in order to allow message passing within this directed acyclic graph, we create a parent is a parent part of to sort of reverse the direction of the edges. And after learning that, each Go terms will have a 120 dimension of representation, and that representation contains the hierarchical knowledge of the Go terms. And in terms of the input, we slightly rephrase the geontology annotation as the input of the model. We add some special linguistic markers to split the evidence and the Go terms. And within the evidence, we also use a special marker called Jing to conditioning these evidence text. So to make sure that the input, to let the model knows that the input consists of 3 components: the Jing product, the evidence text, and the Go terms. And after doing that, we embed the previous pre-trained graph neural network model and this input to a PubMed-BERT model. So we can see that at the last layer, we combine the embeddings of CLS tokens with the Go term, the vertex encodings from the GNN. So to ensure that the language models can capture both the semantic features of the Go annotation and also the hierarchical features of the geontology. And because we're modeling the detection of inconsistency as a multi-type classification task, therefore, we put these embeddings into a multi-layer perceptron as a final end-to-end classifiers. And also, we compare this model with several different alternative versions, and also use a concept mappers with some rules to make sure it can satisfy a inconsistency detection tasks. So we compare that as a baseline setting. So we typically focus on 2 aspects of the model's performance. One is the correctness, and the second one is the confidence. I will introduce both of them. Regarding the correctness, we use precision, recall, and F1 score for the detection of each specific type of inconsistency against the others. So these 3 metrics are standard in machine learning classification tasks nowadays. And here are the definition of the true positives, false positives, and false negatives. The F1 score is simply a balance between the precision and recall measure. So for example, if a model predicts all instances as over-specific, it will result with a perfect recall of that type, but with a very bad precision. So F1 score takes precision and recall with half contributions to the final performance scores. And in terms of uncertainties, the quantification of uncertainty is using Shannon's entropy. This is also a standard used in information theories. For example, it assumes that a situation, if there are 2 classes, 2 types of inconsistencies, while the model has a probability of 50% for each one, then this indicates that the model has a strong uncertainty for the prediction. But compares to if a model has 90% of probability towards a prediction and only 10% towards the other, this indicates that the model has a strong prediction confidence. So this is the measure or the ideas of entropy. We assume that our model shall be correct and also confident in its prediction. So based on this, when a number of suspicious records is forwarded to human curators, it is less likely to be overwhelming to the curators. And for this slide, it shows a table of results directly extracted from one of our publications. The key indication of this slide is that we find automatic models can capture the hierarchical knowledge in the geontology. And this knowledge is very important for distinguishing the small nuance or small differences in the semantics of Go annotation. And also, we find the model is robust to out-of-distribution instances, which means it can support one of its feasibility aspects in the real-world setting. And this slide is much straightforward in demonstrating the performance of the model. We apply this model to a real-world database, which is the Jiyu provided by the NCBI. These are the two inconsistencies identified by the model. I cannot guarantee if my interpretation is correct, but I assume for the first instance, it is flagged as overspecific, probably because the evidence sentence uses hedges or cautious words. And the second one is flagged as overbroad, probably because the model thinks that a better Go term can be negative regulation of multicellular organismal process. So moving to other aspects we have just introduced, we find the model is robust to noise within the training set. We can see that it is still able to maintain a decent performance and metric score when we fine-tuning it with over 40, like less than 40% of the noise. And also here we show how does this model perform, how performance changes when we toggling its uncertainty threshold. So it means when we increase the uncertainty threshold, more instances will be considered as positives during the F1 measures. So that is why when providing uncertainties on the x-axis, the y-axis, the F1 scores changes. But unfortunately, we find that we still need to find a better way to maintain the correctness, which is the F1 score, when setting it to a high confidence mode. However, in overall, the encoding of the Go hierarchical knowledge is helpful to maintain the confidence. And the last aspect is different from the biological views. This might be interested to some text mining researchers. So we designed some linguistic test suite to see how linguistic features can influence a model's performance. So these measures are going to quantify some linguistic features in each instance. So a single instance is a single Go annotation records with the input to the models. And we analyze whether the appearance of that linguistic features will influence the model's performance. So here I have just illustrated 3 of the linguistic test suite. So the Go lens, which is the first one, is to measure or to count the number of English words in the Go terms. We measure this one because broad Go terms tend to have a shorter length, such as biological process, it only has a length of 2 English words. While the leaf Go terms, which are deep Go terms in the Ontology graph, they're longer. So the figure on the right shows that if the associated Go term is longer than 4 English words, the model is less confident to predict it as a consistent instance, or more confident to predict it as a over-specific instance. So it means the length of the Go terms actually matters, can influence the model's performance. The radius of the circles indicates the number of instances that satisfying the linguistic features and the uncertainty scores. So, and yes, and there is definitely more to talk about, but I guess I will wrap up here because there will be too much details. So in this studies, we have made a feasible implementation for automatic inconsistency detection for the Ontology annotation. And on top of this, we also found a suitable leverage of Go hierarchical knowledge leads to the improvement for the implementation. And we also consider both correctness and the confidence as 2 most important aspects for the feasibility of such kind of systems. And below, I listed 2 of our related publications. The first one is our most recent publication to the ISMB 2022. And we have also published an early exploration paper in the BMC Bioinformatics in 2021. And in terms of future directions and opportunities, here are some takeaways. Firstly, we think it is worth to study some inconsistency detections by using related background knowledge. As we have just explored, the Go hierarchical knowledge is one type of the related background knowledge, and it significantly contributes to the improvement. The second one is we can seek ways to extend the detection of inconsistencies from a single Go annotation record to interconnected Go annotation records in the databases. The third is currently we're just covering Go annotations with direct literature support, meaning they have experimental type evidence code. But in futures, maybe we can extend this detection after we have leveraged the related background knowledge, such as we can extend it to support the detection of traceable author statement or annotations that are inferred by the curators. And finally, we also hope that when the model becomes more successful, we can use this model, take the model and apply it to a real-world databases and help curators to support a large-scale Go annotation database quality analysis grounded at the record level. And finally, I would like to state the acknowledgement to the contributors to this study. The Justin Zobel, Nicholas Giat, and Karen Verspools are all my supervisors at the University of Melbourne. And Karen Verspool is also the professor from the RMIT University. Benjamin is one of my colleagues at the University of Melbourne. And I would also want to thank the International Society for Biocuration. They have provided me valuable feedbacks when I'm trying to design heuristic rules in simulating real-world inconsistencies and help me validating or assuring that the generated instances are meaningful, sensible, and truly valid in the real-world databases. Thank you. So this is my talk.
Thank you so much, Jiyu. Impressive talk and work behind. Congratulations. I'm wondering if there is a question from the audience. So let me check in the panel. I cannot see any question yet, but I have some questions for you. Yeah. From a non-expert text miner, so I'm sorry from the beginning. So first, I'm interested on how did you generate the synthetic data? Because I'm also on the issue of generating synthetic data for machine learning. So if you could expand more of what kind of heuristics or how did you do this?
Yeah. Let me go back to that slide. So like this one, so when in terms of generating inconsistent instances, we often need a seed for that generation, and that seed are consistent instances. And what we're trying to do is just to modify part of the consistent instances in a way of simulating inconsistencies. So for consistent instances, we sample records from the biological databases if they have experimental type evidence code, because these part of the instances are considered as most reliable. And of course, as I mentioned, there are noises within the dataset. So for here, we're just assuming that it is a silver standard dataset. We consider all of the seeds are truly consistent. And then based on each different type of inconsistencies, we stick to their characteristics and design rules to modify part of that. Like the overspecific type inconsistencies, it shows because the selection of the Go terms is overspecific. So to generate that, we simply find a overspecific Go terms to the original Go terms in that Go annotation in a way of simulating over specific type.
And I am understanding correctly that you introduced inconsistencies, you created inconsistencies, and based on real biological databases data, and also noise, right?
Yes. Yeah.
And can you define noise? I missed that. For sure you define it, but again.
Yeah. Let me find. Yes. So a noise is like within real world databases. I guess most like major parts of the instances within the real world databases are consistent because they're already manually curated. But some of the instances are not. They're inconsistency, which needs to be identified. But now, because we are like treating them as consistent, we're assuming them as consistent as a silver standard. So these inconsistent instances will be seen as noises. But the challenge is we do not know what is the scale of noise within the sampled data or in the real world databases. So instead of trying to like hire curators to quantify the scale of the noise, we're trying to sort of simulate the different scale of noise and see how different scale of noise can impact the implementation of our systems. So, yeah.
No, go ahead, please.
Yeah, so as a result, we can sort of have a performance window to see what would be the best performance for the models and what would be the worst situations when there are too much noises within the training set.
And to define this, you said that curators help you. So you add people in the loop to generate the synthetic data?
Yeah, so far it's because annotating like gold standard data set is very expensive. Currently, what we're doing is we're just trying to make sure that the heuristic rules we designed is meaningful, it's sensible, and also we just randomly selected a few generated inconsistencies and send them to the curators to see does that make sense to them. So in a way of sort of as a qualitative case evaluation.
Understood. I can see some questions on the Q&A panel. So first question from Tiffany Calhan. Do you think the work that you have done on the Go could transfer to other ontologies that have annotations? For example, the human phenotype ontology?
Yes, that's a very, very good question. We do believe that it is very possible to extend this implementation to cover other ontologies because the core of these systems or the framework is pretty much the same as long as it is required to verify or make consistency inference over textual evidence or textual scripts, then it shall be extensible.
Great. And another question from RJ Murray. Are there instances of conflicting assignment of gene annotation, i.e. group A annotates one way while group B annotates differently? Though these different definitions simply add noise, or are there specific controls that are in place to avoid such confusion?
Sorry, I don't actually really understand that question. You mean the inconsistency? You mean there are different annotations between different groups of the annotators?
So I don't know if said we can invite the attendee to speak out himself or herself.
Let me bring down the screen.
In the meanwhile, yeah, can you read the question to you?
Yes. Do the same genes get annotated differently? Yes, actually, there is one type of inconsistency we covered, which is the irrelevant gene mention. But I guess we're going on a different direction. That is where there are, for example, there are two genes mentioned in the same publications, but the experiment is actually targeting only one of the genes, and the other gene is just a comparison, or simply the experiment is on a mouse gene, but the comparison is for a rat gene. So that is the type of inconsistencies that we focus on. But you have a good mention. Yes, the same genes get annotated differently across different groups. Yeah, maybe we shall also include that in our design.
I have another question. So you mentioned that you capture the semantics and the hierarchy of Go through the embeddings.
Yes.
Could you further explain how?
Yes. So let me go back. Yeah, so for this slide, so firstly is we can actually download a Ontology graph from the Gene Consortium databases. And currently we only include two types of the edges, which are ISA and part of, because these two edges describe the specificity relation or hierarchical relations between different ontology terms. So it will be nodes as Go terms and edges as within this graph. And then we design this graph neural network models. And we hide all of the edge types between the nodes. We just provide the graph with the text attribute of the Go terms. And we let the models to infer what is the relation type between a pair of Go terms. So, for example, if a feeding behavior and suckling behavior is treated as input to this graph neural network models, it shall be able to predict that one is a suckling behavior is a feeding behavior, and feeding behavior is the parent of suckling behavior. So after doing that, there are some iterations within this graph, and it will result with an embedding for each Go terms. And this embedding will be 120 dimension long. This representation, and this representation has the encoding of the hierarchical knowledge of the Go terms.
Okay. And I have another question. So it seems to me that the quality of the evidence sentences can play a major role in the quality of the prediction to detect these inconsistencies. What is your opinion, and how could be improved in order to detect inconsistencies better?
Yes, yeah, that's a very good question. So as we mentioned, we tried with two different types of data. One is the BC4Go corpus. It has a very high quality evidence because they're all grounded at sentence level. But for the second part, we do not have curators to really highlight or extract that evidence at sentence level. So we simply just input the entire title and abstract to the models. So yes, the performance drops a little bit compared to the BC4Go corpus, but because the quantity of the input data is much larger than the BC4Go, it's still able to maintain a decent performance. But we strongly believe that if we can implement a model running ahead of this inconsistent inference model to firstly to identify evidence sentence from the full text literature, it shall be make significant contributions to improve the quality of the evidence and improve the model's performance in overall. Currently, we're also testing with the GenRef, which are short statements created by the curators. And yes, we do find that it performs better than just inputting it with title and abstract. Yes.
Great. And my last question, you are early career researcher in your third year in your PhD. Do you have some advices to more younger, just starting off PhDs programs? Please, what would be these advices?
Yes. Yeah, thank you. So, so I guess it's very special for me and most of the students that's starting at my age. I started my PhD at 2020 February, which is just 1 month before the impact of pandemic. So most of my courses stayed out of the lab, out of the office, and stayed at home. I guess for me, the most important thing is just trying to get involved with the community, get involved with others, and do not be influenced by any quarantines and so on, because it is very important to share your ideas or absorb ideas from your peers, especially after you have, at the beginning, you have done lots of literature reviews, you have lots of thoughts. It is more important to have a little bit discussions with other people's. So it can help you to clear out or make out, to clarify like which direction is appropriate to go with. And also, I think it is very important to have a like meaningful conversation or communication with your supervisors, because sometimes you may get tracked into a rabbit holes. You go in too deep into a certain small problems and cannot get back. Your supervisors are very experienced, and if they spotted that, they can just pull you out and make sure that you're still on the right track. So you don't have to really waste too much time on a small, deep problem.